Greg Rand. I work with Noble Capital Markets. Just quickly before I introduce the company, I'd like to handle a couple housekeeping issues. At the end of the presentation, we'll open up for Q&A. I'll direct the first question and then open it up to the audience for their questions. Of course, be aware that there's also a breakout session immediately following outside these doors here with the company, so you can follow up with them at that juncture. And with that, I'd like to introduce David Mazzo, who is the President and Chief Executive Officer at Lisata Therapeutics. David, welcome. Thanks for coming.
Thanks, Greg. Thank you, everyone. Good morning. Thanks for getting up and coming here. It's really a pleasure to have the opportunity to introduce you to Lisata Therapeutics. So like all of the other public companies here, I'll remind you that I'll be making some forward-looking statements during the presentation and ask you to please keep those in mind as you make investment decisions. So I'd like to start with an overview of what Lisata is. So we are a clinical stage therapeutics development company that's working on a novel solid tumor targeting and penetration technology to improve the efficacy of anti-cancer drugs. So our product is an adjuvant. It is co-administered with other anti-cancer drugs. It does have some specific anti-cancer activities, which I will describe momentarily, but principally it's designed to work in concert with other products. Like many companies, we have a seasoned management team. We've got over 100 years of successful international drug development experience amongst us. Of course, our products are protected by intellectual property, but unlike a number of other companies, we actually project quite a few milestones, both data and business milestones over the course of the next 12 to 24 months, any one of which could be value creating, and the accumulation of all of that news should increase value for the company and the product significantly over time. We also have a number of partnerships with names that I think at least some of which you'll recognize, and that serves to, in inverted commas, validate our technology as others have looked at it and believe in the promise as well. But perhaps in this difficult financial environment, the most important thing that differentiates Lisata is the fact that we are well funded. We have cash through the beginning of 2026. All of our development programs are funded through data, and I think that's really an important aspect at the moment. So let me talk a little bit about what we're doing and why we're doing it. So we are a company working on cancer, and everyone knows that cancer remains a major problem. It still is the largest killer of people in the developed and even in the emerging market world. But what they don't know, I think, is that most of the newly diagnosed cases of cancer, about 90%, are actually solid tumor cancers. And these are the cancers whose names really sort of, strike fear into the hearts of the patients. It's pancreatic cancer, it's bile duct cancer, ovarian cancer, brain cancer, etc. These are cancers that are very difficult to treat and today have, unfortunately, a very poor prognosis for the patients who have them. Now, the reason that the current treatment paradigms are suboptimal is that these tumors have a physical barrier around them. It's a biological barrier called the stroma that's comprised of tumor endothelial cells, tumor cells themselves, fibroblasts, a number of other mixed cellular matrix. And they actually prevent products, anti-cancer agents, from penetrating the tumor and actually doing the job of killing the tumor cells. In addition, these tumors have a microenvironment, that's the biological environment immediately proximal to the tumor, that is highly immunosuppressive. So that means that the tumors are in the lingo, they're cold tumors, and that means that your own immune system is not doing very much to suppress the cancer. And you can't get around this biological barrier and this immunosuppressive microenvironment by simply prolonging or escalating the dose of common agents, because you end up with off-target side effects. And the other part that makes treating solid tumors difficult is that for years it's been a real challenge to translate the results from animal models into humans. And I think you've all known we've cured pancreatic and other cancers in rodents hundreds of times in the last decade, but it hasn't been able to translate to humans. So we're working on a technology that leverages a natural existing transport mechanism. It's called the C-end rule or CendR transport mechanism. and we use that to target the penetration of the anti-cancer agents into the tumor, making them much more specific. Now, there are many targeting agents that are available, and RGD peptides are just one example of them. What we've done is we have taken that to the next generation, and we are using internalizing RGDs. And these combine both the targeting to the tumor and the penetration capabilities. LSTA1, or certepetide, our lead program, is an internalizing RGD peptide that exploits the C-end rule of active transport mechanism that targets these solid tumors, and it is in clinical development as a tumor targeting and penetration enhancer. And as you'll see, we're making great progress in that regard. Now, LSTA1 converts that stroma that I described earlier from a barrier actually to a conduit for anti-cancer drugs. And what makes this really promising is that it is agnostic to the modality of the anti-cancer drugs that are being co-administered. That means we can mix and match with chemotherapies, immunotherapies, radiotherapies, antisense therapies, etc. It also is important to note that LSTA1 actually combats resistance and metastases. So we have data that shows that it selectively depletes the immunosuppressive T cells, enhancing the cytotoxic C cells, and inhibits the metastatic cascade. So it does have a synergistic effect with the anti-cancer agents with which it is administered. Now, we're developing this product in a two-pillar strategy. On the one hand, we're pursuing a rapid global registration in pancreatic ductal adenocarcinoma, or MPDAC. And here we are in phase 2B, and in fact, the phase 2B program is almost completely enrolled. You'll see a notice to that effect coming out imminently. We're starting with a combination of one of the two standards of care in pancreatic cancer, which is gemcitabine plus nab-paclitaxel, but you'll see we actually have programs working with the other standard of care for MPDAC, which is FOLFIRINOX, and I'll talk about those momentarily. Now, in parallel, we're running a number of proof of concept studies with a variety of different standard of care regimens in a plethora of solid tumor cancers. And this is to demonstrate the broad utility of LSTA1 and its ability to be mixed and matched with all types of different agents. And you'll see that we have a number of programs underway in that regard. So as I mentioned earlier, we have a number of partnerships that have existed already. On the R&D side, we've partnered with clinical research groups like the Australasian Gastrointestinal Testing Group out of Australia and New Zealand, with WARP9, which is a foundation supporting GI cancer discovery and treatment. That's also in Australia, and with Roche, where we're part of their Morpheus platform, looking at combinations of atezolizumab with a variety of new agents like LSTA1. Additionally, we have a strategic partnership with Qilu Pharmaceutical, a large pharmaceutical company in China. They've licensed the exclusive rights to LSTA1 in China, Taiwan, Hong Kong, and Macau, and they've assumed all the development, commercialization responsibilities and cost in those territories. There is a joint steering committee, however, which we sit on and chair, and it allows us to ensure that their development efforts remain consistent and in sync with the global development of LSTA1. And we've collected a number of major milestones from them already. $10 million up front, $5 million when they filed the first IND. We have millions of dollars due to us in the coming years based upon completion of the technology transfer of manufacturing into China, and ultimately when they start a phase 3 program, get approval, and of course we have commercial milestones based on sales. And as I mentioned, because we can mix and match here, there are a number of additional partnership opportunities that exist for many combinations with LSTA1 in a variety of different indications. So let me tell you a little bit about how this actually works. LSTA1 is a 9-amino acid cyclic peptide, and it's been designed to have an extremely high binding specificity and affinity for alpha V, beta 3, and beta 5 integrins, which are proteinaceous receptors which are upregulated significantly on tumor cells and tumor endothelial cells on the surface of the stroma. Once LSTA1 is bound to these integrins, it's susceptible to proteolytic cleavage by protein enzymes that exist in the tumor microenvironment, like furin and others, and it gets split into two different linear peptides. The 5-amino acid linear peptide is actually a C-end rule peptide fragment. And it has a very high binding affinity for an adjacent receptor called neuropilin-1. And when neuropilin-1 is activated, the C-end transport pathway is initiated. And it's manifested by the formation of small vesicles at the surface of the cells, which encapsulate anything in the circulation system, like co-administered anti-cancer agents, and it percolates them through the stroma and deep into the tumor where the payload is deposited. What's also noteworthy is that when the C-end rule transport pathway is activated, the gap junctions, that is the spaces between the cells in the tumor and the stroma, relax, and that allows for increased extravasation of immune cells into the tumor. And so that helps your immune system, in fact, contribute to fighting the tumor. Now, we've got a number of clinical and preclinical data here, and I'll just show you a few examples. In this particular preclinical model, which is a nude mouse which has been implanted with a human pancreatic cancer model, we're using something called fluorescent quantum dots to demonstrate the selectivity and specificity of LSTA1 in combination. So when you simply dose the fluorescent dots to a mouse, if you didn't apply an etching solution, the entire mouse would light up under the fluorescence, the fluoroscope. But when you apply an etching solution, anything in the circulation is quenched, and so you don't see it. So that's why we have the dark mouse on the left. If we do the same experiment, but we add LSTA1, LSTA1, based on the mechanism that I just described, takes those fluorescent dots and focuses them entirely into the tissue of the pancreatic cancer. And then we apply the etching solution so that everything in circulation is quenched, and you can see the only thing that lights up is the pancreatic cancer tumor itself. Highly specific, highly predictable. And then this is a sampling. I don't expect you to read these, but there's a bibliography on our website. There are more than, it's now approaching 350 publications in peer-reviewed journals that demonstrate that LSTA1, when combined with a variety of different types of anti-cancer agents and a variety of different solid tumor preclinical models, consistently improves the efficacy of those anti-cancer agents in those models. But what's really most important is what happens in people. As we said before, the translation from animals to humans is always not easily predictable. And so here we have a phase 1b/2a program that we conducted in Australia. It was in first-line MPDAC patients, 3 sites in Australia, 31 patients, 29 of which were evaluable. And as a phase 1 trial, it was showing that LSTA1 was highly well tolerated. There was no dose-limiting toxicity. And importantly, the safety of the combination of LSTA1 and the chemotherapy, which in this case was gemcitabine nab-paclitaxel, was identical to the safety of the standard of care alone, meaning LSTA1 did not augment the adverse effects whatsoever. But safety is great, but what's really important is efficacy. And look at the astounding results that we've demonstrated in this small trial here. In fact, the principal investigator calls this a breakthrough. He's never seen anything like this, and he really believes that this could really change the paradigm for treating these difficult-to-treat solid tumors going forward. The efficacy results are probably more easily seen visually on these bar graphs. So you can see, first and foremost, what do cancer patients care about? They don't care about biomarkers. They don't care about, response rates. They want survival, right? And so here, for these patients, we've been able to improve the median overall survival by more than 55% and the progression-free survival by greater than 75%. What's also important in clinical trials is consistency. You want to see that all of your endpoints and markers are moving in the same direction, right? So there's no anomalies and there's no confusion. And here you can see complete response, partial response, objective response rate, the disease control rate at 16 weeks, as well as the relative change in one of the most important cancer biomarkers, CA19-9, all show vast improvements in the presence of LSTA1. So this is really highly promising, and this led us to embark on a much stronger program, a broader program in MPDAC. Now, you should know, before we talk about the clinical program specifically, the regulatory strategy here has been, of course, to also exploit all the benefits that a breakthrough in these cancers could bring. And for that, we've already achieved a fast track designation for pancreatic cancer in the United States for LSTA1, as well as orphan drug designations for pancreatic cancer in the United States and Europe, and an orphan drug designation in the United States for glioblastoma. We'll have several other applications in for different cancers in this regard. And these bring, for those who aren't familiar, certain benefits. Of course, as the name implies, Fast Track gives you a faster review and more frequent communication, but importantly, this makes you eligible for accelerated approval, and that's really important for these solid tumor treatments. Orphan drug designation, of course, gives you certain tax credits, makes you eligible for grants, but most importantly, extends your market exclusivity significantly. And so all of these have commercial as well as regulatory value. What happened here? We're stuck. All right. Why are we not moving? Okay, there we go. So now we have, the next two slides are just tables that demonstrate the number of programs we have in place, where they stand generally in terms of enrollment, and also where they're being conducted. And you'll notice that this is a very large table of clinical trials for a small company like Lisata, which is only 25 people. We're able to do this and afford to do this because we have a very capital-efficient development plan. We're doing our studies in selective geographies like Australia, where we get R&D rebates that help reduce the cost, as well as other countries where the costs are much lower, but where the GCP, the good clinical practices, are quite high. And we're also combining with a number of those partners that you saw listed on earlier slides, who either contribute or in some cases completely pay for the trials, but we haven't had to give up any commercial rights in any of those cases. So it's a very capital efficient approach. All of these trials on this page and the next will be leading to data, and I'll show you a visual that gives you some sense of the timing for that. And that'll be starting in 2024. But the most important, I think, is to note, which is the top line here, which is the ASCEND trial. That's the phase 2b trial, 155 patients, blinded, randomized, placebo-controlled trial being run at about 40 sites throughout Australia and New Zealand. That trial, as I said, is almost completely enrolled, and we expect to have data from that trial, which we believe will be seminal to the program, at about this time next year. So there is really an important data card that will be turned in about 12 months' time, and all of these other programs will be supportive. And you can see there are trials in pancreatic cancer, the BOLSTER trial, the second one here is being done in cholangiocarcinoma, which is really bile duct cancer, as well as head and neck cancer. We'll be looking at possible programs in osteosarcoma, and we have a program in glioblastoma, a program with the other standards of care in pancreatic cancer, with and without immunotherapy, etc. And we're also looking here at a program in peritoneal carcinomatosis, as well as programs that look at appendiceal, colon cancer as well. So a lot is going on here, and a lot of news flow will be coming out in due course. And speaking of news flow, I can give you two slides that give you the visual here. Now, one of the things that's important is our original projection for enrollment in ASCEND was that we would complete enrollment at the end of the second quarter of next year. We're quite ahead of that, as I said, and you'll see an announcement sometime soon that will talk about the completion of enrollment. So we're about 6 months ahead of that, and we will have the data from that first cohort at this time next year, so very important data points. And then you can see all the other trials where you have data points, and what's not shown here is the possibility of business development deals that will become more likely as we show data from some of these trials and as we approach ASCEND. Financially, our company is quite stable. We just filed our third quarter quarterly statement. We have about $55 million in cash on the balance sheet. We have no debt, and we have cash projected through the first quarter of 2026. It's a very conservative investment, and all of the programs that you saw on those slides are funded through data, which is quite important, and our cap sheet is, our cap tables are quite clean. So just to summarize, we believe that there's really a strong investment rationale for our company at the moment. While we are stuck in the doldrums like many microcaps in the healthcare industry, our fundamentals are really quite strong. We're financially stable. We have data coming out. We have strong technology with intellectual property support, strong people, and strong partners. So we think all of those combined make for an interesting investment thesis for Lisata, and I hope that you'll consider becoming a partner with us as we work to cure a number of these difficult-to-treat solid tumor cancers. And I thank you for your interest this morning, and I look forward to answering some questions.
Thank you, David. Appreciate that indeed. Thank you very much. Let me start with the first question, and I'll open up the audience if you don't mind. David, you talked on the resistance aspects of the therapy. How does it reduce resistance at the same time enhancing cytotoxic behavior on the tumor?
Well, it does so in two ways, and they're really separate approaches. So the enhancement of the cytotoxic behavior is really by opening, if you will, the door into the tumor through that active transport mechanism and allowing those anti-cancer agents to not get hung up in the stroma, but to actually get into the tumor. The way that it enhances or prevents metastases or helps to prevent resistance is by its effect on the tumor microenvironment, where it makes these cold tumors hot. It makes them much less immunosuppressive, allowing your innate immune system to contribute, and any, in fact, dosed immunotherapies to contribute to the efficacy of the treatment.
Okay, thank you. That opens up the Red Sea kind of thing, part of the Red Sea kind of thing, and allows everybody to pass through. That's excellent. Anybody have a question out here? I saw hands. Go ahead.
Yes, I was just going to ask you, assuming things go well and you get either an accelerated or approval, do you plan to use, combine your resources with a major company for a sales expansion with their sales force? Are you going to hire your own people? What are you going to do to get it through after it's approved?
So I thank you for asking that question. One thing I should mention, we've already had a number of discussions with regulatory agencies about the ASCEND data. And the first one, of course, would be Australia, New Zealand. And there, the regulatory authorities have already told us that if this data is positive, that in combination with the phase 1 data, they would consider it acceptable for an accelerated approval application. So that's a good. We'll have similar conversations with FDA and EMA coming up. Now, all companies need to plan for success. And so the stock answer I should give you would be that we'll build our own sales force and we'll plan to do that. The reality of the situation is that that's likely not to happen. If our data is positive, the product either will be purchased or the company will be purchased, and I think that'll be just fine for our investors.
Okay, thank you.
Thank you. Another question?
Yes, I had a question regarding the photos you showed of the mouse before and after you had the LSTA one attached to it. When we're talking about the cancer medication, a lot of the things you have is that when they give it to you, they're not just attacking one piece, they end up attacking the whole entire body. So if I'm understanding what you presented correctly, that means that LSTA basically takes all that cancer medication that is in your body and focuses really on the tumor, in that sense. So it's not like it's everywhere else.
Well, what it does is it takes of course, so let's think about how things are administered. You'll have an IV set up, and you'll have the chemotherapy or the immunotherapy being introduced. You would dose through the same IV, LSTA1. LSTA1 then activates this CN rural transport mechanism. So the chemo or immunotherapy is in the circulation, but imagine as it's in the circulation, every time it passes through the cancer-laden area, it gets sucked in.
Okay, that was my thing, because that's a big thing that when you have chemo or anything else like that, that you become completely sick,
Right.
because they're not necessarily just focusing on this point. So that means that it really does filter, it really focuses on those tumor cells,
That's
and that's where it goes to. It doesn't end up in other parts of the body.
That's it, right. And so that gives you one of two opportunities here. On the one hand, you could be in a position where you get much increased efficacy, as we've demonstrated, with a similar safety profile. Or if the patient can't tolerate the chemo or immunotherapy, you could potentially reduce the dose and still get a reasonable amount of efficacy, but less side effects. So you have this new tool to balance.
I think we're out of time,
Well, he
unfortunately.
had one question out there.
Yeah, 30 seconds, basically, sorry. Go ahead.
Can you say something about your patent situation? Your patent.
Oh, our patents, yes. So our patents cover the product currently out into the mid-2030s. We have patent extensions that are being prosecuted now, which would
Do
take
you
us...
have composition of matter?
Sorry?
Do you have a composition of matter?
Yes, we do have composition of matter.
Of this?
Yes, of this IRGK certificate.
And what's your head count? What's your current head?
25 people.
Thank you. I think we're out
We
of
have
time.
a breakout room
Unfortunately-
if anyone would like.
Yes, there's a breakout session outside.
With more than 6,000 small and micro-cap companies listed, if you're looking for the next Apple, then ChannelCheck truly is your tree. It's an investor community where you can access independent Wall Street research, advanced market data, balanced news, corporate videos, and podcasts. You can even register to attend in-person Meet the Management Series lunches, and to be part of the preeminent annual Small and Micro-Cap Investor Conference, NobleCon. And here's the best news. Since ChannelCheck is sponsored by the featured companies, premium membership requires no investment. It's absolutely free. So check it out. Log in today. Find out why ChannelCheck is North America's leading source of research on the companies that just may change the world.